What if you had a tool that could do almost anything, from opening a letter to removing a splinter and even filing one's nails? The Swiss Army knife is a versatile multi-tool pocket device. Now, imagine a specialized Swiss Army knife, one that could slice more than just fruit. A Swiss Army knife that could fix your genes. CRISPR. Just like a Swiss Army knife can fix a bike, cut a rope, or open a can, CRISPR can cut, edit, replace, or disable the genes of different organisms. Scientists choose the blade corresponding to their task, and despite its simple system, CRISPR is powerful enough to change life on Earth. It is small, yet it holds many functions, whether in your pockets or in your genes. CRISPR, or clustered regularly interspaced short palindromic repeats, is a technology first discovered in bacteria. In 2012, scientists Jennifer Doudna and Emmanuelle Charpentier paired it with an enzyme called Cas9, leading to a revolutionary discovery. In short, CRISPR uses the Cas9 enzyme and a guide RNA to target and remove specific DNA segments, so it can mutate any gene. Normally, it's used in utero or shortly after birth. So, how does this work? As described by the Stanford report, here's the short answer. When a virus infects a bacterial cell, CRISPR helps establish a memory. The bacterium takes a piece of the virus's genome and inserts the DNA into its own genome. From that newly acquired DNA sequence, CRISPR creates a new guide RNA sequence that helps CRISPR find the invader via sequence complementarity. So, the next time when the virus infects that bacteria cell, the guide RNA rapidly recognizes the virus DNA sequence, binds to it, and destroys it. In other words, scientists design a guide RNA to match the DNA that they want to edit, which leads Cas9 to the target sequence. Cas9 cuts the DNA, which the cell then tries to repair. This is when scientists insert or delete specific genes. CRISPR, as put by Dr. Doudna, offers a way to rewrite the code of life. The first CRISPR-focused treatment was in July 2019. Victoria Gray was the first person in the U.S. to receive CRISPR-based cell therapy for sickle cell disease. Doctors removed her bone marrow cells, edited the faulty gene that caused her disease, and reinfused the edited cells into her body. Within months, her symptoms faded. Now Gray remains healthy as a result of CRISPR. It's already being used to treat genetic diseases like HIV or hereditary blindness. Its impact, however, goes far, far beyond medicine. Agriculturally, CRISPR yields a multitude of benefits. One of many examples is that of mushrooms that don't brown. In 2016, the USDA approved CRISPR-edited mushrooms designed to stay fresh longer and reduce food waste. Similarly, CRISPR has edited rice and corn crops, increasing drought and pest resistance. This falls back into selective breeding, a commonly used tactic in plants and animals. It involved choosing parents with desirable traits, breeding them with each other, and producing better offspring. Finally, in terms of the environment, CRISPR has already made significant changes. In Africa, for example, mosquitoes are being genetically modified to block malaria transmission. On the contrary, CRISPR is also being tested in reviving genes as to protect coral reef and honeybee populations. In the future, CRISPR could someday be used to prevent diseases that we once feared. Crops can be tailored to their climates, and different animal species can adapt to climate change. Of course, as the Spider-Man movies put it, with great power comes great responsibility. CRISPR can't always be precise and sometimes doesn't cut the right spot, possibly causing dangerously mutated genes or even cancer. Furthermore, the first approved CRISPR cell therapy, Casgevy, currently costs a whopping $2.2 million per patient. What's the use of such a helpful tool if it's open to a limited audience? Ethically, CRISPR has been debated for years. It could be used to design children with certain characteristics, causing practices where specific traits are favored over others, and people could enhance traits beyond disease. This could lead to some sort of a social hierarchy. Apart from that, CRISPR can help plants grow more easily, but doing so can tamper with ecosystems. This could disrupt pollinators or even create new allergens. With CRISPR, modified genes could spread, and editing the DNA sequences of different species might reduce natural diversity. CRISPR holds the power to change medicine, agriculture, and, as it happens, evolution. As biochemist Feng Shang says, We need rules to make sure it's used to benefit humanity and not harm it. In a nutshell, CRISPR is rapidly making changes to the natural world. It's up to us how we use it. Thank you.